Assalamualaikum warahmatullahi wabarakatuh.
Folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand because there are different ways of folding that can happen in the different structural levels. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, exposed to high heat, for example, you can disrupt the interactions that we had talked about taking place at the different structural levels. This can denature the protein, which disrupts its shape. This prevents it from functioning correctly, and depending on what caused it to be denatured, sometimes you are interfering with many levels of protein structure. Sometimes it's just one or two levels. Sometimes denaturing a protein may be reversible, but in many other cases, it's not. The environment that a protein is in definitely matters for its functioning.
Take 1 milliliter of test solution in dry test tube and another tube take 1 milliliter distilled water as control. Add 1 milliliter of burette reagent to our test tube, which is 0.1 molar sodium hydroxide and 1% of copper 2 sulfate. Purple color solution indicates the positive result, which means the presence of protein.
Add 2 ml of albumin in 3 test tubes. Add 4 drops of copper sulfate in the first test tube. In the second test tube, add 4 drops of lead acetate. And mercuric nitrate into the third test tube. Then, swirl the test tube. Add another 5 drops of copper sulfate. Observe the change.
So I will show you guys the procedure of precipitation of protein by acids. First of all, the experimental pipette 2 ml of protein solution, which are albumin, casein and peptide into each test tube. And then 5 drops of tannic acid are added to each test tubes. And then the test tube are swirled, the observation are recorded, and then the 2 ml of 0.1 molar sodium hydroxide slowly added into each test tubes, and then the observations are recorded.
Okay guys, so this is the result.
Pipette acetate acid and then sodium acetate. Add 1 ml of casein into each test tube. Observe the changes after 30 minutes. Observe the changes again